Good evening. Welcome to the How To Academy on Tuesday evening. And I'm always fascinated by the people that I meet doing these broadcasts, but this is a particularly great evening because I imagine many of you watching at home have friends who it's turned out have suddenly become experts on vaccination. And so you will need this conversation because tonight we actually have people who really are experts on vaccination. They are authors of this fantastic book, Vaxxers, Professor Sarah Gilbert and Dr. Catherine Green, who developed the AstraZeneca vaccine, which, if you are like me, you will have received, or maybe on one, or as I have on 2 occasions. Don't tell my age on that. I've just revealed my age. I had 2 vaccines quite early on. Terrible business. That's how old I am. But we are going to be talking tonight about AstraZeneca, about viruses, about the development as well, and understanding of COVID-19. And if you have any questions that you would like to ask, so I imagine there are many of you with a lot of questions, then just send them in. I will see them in the Q&A, and we will start in the second half of the show dealing with some of your questions. So good evening, Catherine. Good evening, Sarah.
Good evening.
Now, I wanted to go right at the start. There's a wonderful story, Kath, where you talk about being you're out camping, waiting for a pizza. And you have an experience. I wanted to start on this because you had a moment where you talked to someone who just started to say, oh, I'm worried about this vaccine because of them and what they do. And you had a moment of basically saying, well, I am them, and had a chance to, and I wondered how you have felt about the ability of being able to communicate to people that this idea that people who develop vaccines are some kind of, dystopian image of some ogre. Have you found that this discussion has got better in the last few months, especially as the vaccine rollout's been happening?
I think it has. So yes, this was back in late July, early August last summer, and I'd managed to get a weekend away with my daughter to Snowdonia, peace and quiet, beautiful, tranquil, thinking I'd have a couple of days off from the stresses that were filling my daily life at that time. And the lady in front of me was just reiterating some of the perhaps more strange conspiracy theories that were circulating at that time. And I got the feeling that, yeah, one of the things that she was concerned about was that she didn't have knowledge about who was making the vaccine, how is it made. She didn't have a solid foundation which would put her fears to rest. So I just stepped in and said, hi, I'm Kath. I know you're saying you don't know what's in the vaccine, you don't know how it's been developed so quickly, but weirdly, I am the right person in the world to tell you that because it was my team that actually made it, that first dose that went into a human being. I can tell you exactly what's in it, and I'm happy to try and explain that to you, just to give that amount of information that everybody needs when something is new. And that perhaps planted the seed that Sarah and I maybe could do something in order to try and get the information out to the public in an accessible form of who we are, what we did, so that people can make their own informed decisions. I think that's all we can ask, isn't it? That when things are new, we explain them well, and scientists have a duty to do that.
Well, you do explain it well. It's very interesting. And I wondered from your perspective, Sarah, as well, has there been a problem with the fact that actual platforms for evidence-based thinking, for being able to deliver this, that there perhaps aren't enough broad platforms, that sometimes the news media is driven by things which are clickbait rather than evidence-based thinking?
Yeah, I think there's a lot of pressure in trying to get information across in a very, very short form and then shorten it even further to give a headline. So we do try to give interviews where we talk about the science behind what we do, but some of them are necessarily quite short. And then we felt we wanted to write something that really told the whole story, because it needs a bit more time to give the background, to explain who we are, what we do, how we were able to go quickly, and what actually happened last year. And that's why we started on the book.
Well, you say quite early on that back in March 2020, that was in some ways where, then there was a race to find a vaccine. But you also say that in some ways you felt in another way the race had been lost.
Well, I mean, there was times of stress when we could see the pandemic growing and we weren't there yet with getting the project. And of course, all the way through until, later in the summer, we had no idea whether the vaccine that Sarah had designed was going to work. we knew we'd be able to make it, we knew we'd be able to trial it, but we didn't actually know, because it's science, yeah, you don't know the answers until you get the answers. So I mean, there were a lot of stressful times when we could see this disease taking off around the world, and we didn't know whether our vaccine or anybody else's vaccine were going to be effective. So that was, I mean, that was tough. It was tough for all of us, yeah. We were all in the same boat at that time. When I was on James O'Brien on end of April, he said, which was the day we started the first clinical trial in Europe, he said, we've brought a ray of hope, but it really was only hope. We were hopeful, we were confident as far as we could be, but we didn't know this was going to work, remember?
Are you still surprised by that? I remember talking to someone, I think it was April last year, who was based up at the Crick Institute, and they said, you know what, maybe by December we'll have something that is about a 50%er, that it'll be about as effective. And then when I spoke to him, you know, towards the end of the year, and spoke to other people, it seemed there really was, in terms of a success story, this does seem quite remarkable.
Kat? Yeah, it has been very successful. We're now getting what we call real world effectiveness data. So that's not the data that comes out of clinical trials. It's the data that comes out of using the vaccine across the population in real life situations, starting from people who are over the age of 80, because they were the ones who got it first. And so we started to see that a single dose of the vaccine, either the Pfizer or the AstraZeneca vaccine, was keeping 80% of 80-year-olds out of hospital. And that's just with the first dose, and then it improves after the second dose. And so there's a lot more data now showing really good effectiveness when it's real world data against the Delta variant, for example. And, you know, Public Health England keep updating all of this. And it's very, very effective. And we're very pleased to see that in the real life situation, because there's always a bit of a concern that you're testing vaccines in healthy people in clinical trials and it's going to make it look good, and then when you actually start using it widely, it just won't be that useful. But actually it's proving to be very effective. All the vaccines that have been licensed in the UK are showing really good effectiveness.
I was interested as well in the book. You talk a little bit about the fact that, you know, sometimes people will say to you, why didn't you see this coming? And you talk about the fact, well, we did. And I'm fascinated in knowing the process when you're working on, so, because this wasn't, you know, you've been working on so many different vaccines at any one time for different illnesses, for different expected outbreaks. How do you map in terms of going, this is what we need to prioritize if there isn't a major outbreak at a time?
Well, it's all about developing the technology. So what we've been working on for quite a long time is a technology that we can very quickly use to make a vaccine against any different virus that we need a vaccine for. So working on vaccines against outbreak pathogens was what I was doing previously. So things like Ebola, although I wasn't personally leading the Ebola vaccine work, but I did contribute to some of it. Other diseases like Lassa fever virus that causes a lot of cases in West Africa. That's quite important because it results in deafness in quite a large number of people who recover from that infection. Nipah virus in Bangladesh and in India, very high fatality rate. Middle East respiratory syndrome, which spreads in Saudi Arabia. There's also a very big outbreak in South Korea, huge economic impact. So if you're going to make vaccines against lots of different diseases, it's not cost effective to start from scratch each time and, you know, take each disease in turn and say, well, how are we going to make a vaccine against this one? What we want to do is have what we call a platform technology. There's a way of making vaccines where we spend a lot of time developing the technology. How do we make it? What dose do we use? How do we store it? All of those kind of things. And then we can just adapt that to make a vaccine against any of these different viruses that we want to. And it also means that when we have to make a vaccine against a new disease that we haven't encountered before, we can go quickly because we've already done so much of the work just developing the technology itself. So that saves a huge amount of time.
Because that's one of the things, Kath, that fascinates me, is seeing how different the reaction can be in the 21st century than before we were able to. I was fascinated to find out that, you know, you don't need a sample of the virus as long as you have got the genotype. So can you run us through that?
So that's the joy of the method that Sarah and other scientists at the Jenner have been developing over the last 10, 20 years even, is that we need somebody to have got a sample of the virus in the world, because you need to understand its genetic code. But once somebody has isolated the virus from a patient and decoded it, they can just email the sequence to Sarah, and she can use that to design a vaccine. So she can just take, because she'd been working on the MERS vaccine, so that's another coronavirus, Middle Eastern Respiratory Syndrome, and has got really great, I mean, all of publicly available data on clinical trials using that vaccine on the same platform. So the ChAdOx1, chimpanzee adenovirus Oxford 1 platform. So Sarah and Tess knew what they were doing, knew what they needed to do, just needed the code. And once they've ordered the code, then a little bit of manipulation of that code to get it into the sequence inside the laboratory, and then it hands over to me for the manufacturing.
Brilliant. And Sarah, I wonder, but perhaps we can go back a bit, and just first of all, one of the things I think often surprises people is finding out that a virus is not a living thing. Can you explain how a virus works? So in a situation like this, what is going on inside a body?
Well, a virus has some of the characteristics of living things, but not all of them, because we do sometimes talk about killed viruses, killed viral vaccines. So that, by definition, they would have to be live in the first place. So it's a question of how you use the word. But a virus can't reproduce itself on its own, or even with the help of another virus. So a virus is basically a piece of genetic information. It can be DNA, or sometimes viruses are RNA. Our genetic information is all in DNA, but sometimes viruses use RNA instead. And then that piece of genetic information is packaged up inside a shell of protein, which protects the DNA or the RNA and allows it to get inside a cell. And so the viruses that infect humans need to get inside human cells in order to make more copies of themselves. And once they get inside a human cell, the genetic material comes out of the protein shell that's around it, and then it starts to make more copies of the genetic material inside the human cell. And then it uses the machinery inside the human cell to make more copies of the protein coat that it needs. And then it assembles itself into lots more copies of the virus. So viruses don't grow and divide. Viruses infect a cell, they make more copies of the genetic material. That results in making more copies of the protein that surrounds the virus, and then it assembles into lots more copies of itself and then bursts out of that cell. And then all of those new viruses can go on and infect more cells in the same person, causing the spread of the disease through the body. Or they might, if it's a respiratory virus, be breathed out or sneezed out, and then they can go and infect somebody else.
So what's actually going on in terms of, one of the things I found interesting is, with people I've spoken to who've had COVID, is the variety of symptoms that many people have had. You know, one friend of mine just said his hands felt like they were on fire for 2 weeks, and it was just is this common of many viruses, that there is such a variety of different reactions to it? Or is this something in terms of within the coronavirus where we will see these wildly various symptoms?
It's not uncommon to have lots of different reactions to the same virus, because the symptoms that we get after viral infection are not necessarily just down to the virus itself. A lot of it comes from our immune system responding to that virus. So when we get a fever after a viral infection, that's because our body's immune system's kicked into response mode, and we get a fever as a result of that. And some of the other symptoms we get, some of the more serious ones, can actually be when the immune system goes off course a bit and starts attacking parts of the body in a way that's obviously not very helpful. And with coronavirus, which is quite a complex virus, it's not one of the simplest ones, there does seem to be quite a lot of this redirecting of the immune system in ways which then attack the body instead of attacking the virus. So you can get lots of different responses, because it's to do with ourselves and our own genetic makeup, how we respond to viruses, as well as to the virus that we're making that response to.
And then, Kath, so when you start working on a vaccine, what is the model that you work on? What is the starting point? Because I know there's also various different forms of vaccine as well.
So yeah, you can make vaccines in very traditional ways that have been made for hundreds of years, or in the modern ways that we make them today. And so traditionally, you would take the pathogen itself. So in this case, you would take SARS-CoV-2 pathogen, and grow it in the laboratory and find a way to make it safe by killing it, by inactivating and using that as the vaccine. And we have vaccines that are made in that way still today, but figuring out how to isolate and grow a new dangerous pathogen safely and then inactivate that safely takes a lot of time, and therefore might not be the ideal thing to attempt in a pandemic scenario. You can also make vaccines by just using the protein shell that Sarah was talking about. So you could make the protein itself, which won't be infectious, and so it's going to be a very safe vaccine. You can manufacture that in E. coli, say, and use that as a vaccine. So you can have protein-based vaccines. And then, of course, you can use these more modern technologies, which are the mRNA vaccines, which turned out, of course, to be incredibly effective. So that's the Pfizer vaccine, which is delivering instructions to your cells so that you make a little bit of the coronavirus for your immune system to recognize, called the spike protein. And the Pfizer and the Moderna mRNA vaccines do that by delivering mRNA code wrapped up in a package that means it's deliverable to your cells. And ours is an adenovirus vectored vaccine, and that works in a similar way. It delivers a DNA code to your cells, but the vaccine itself is a virus, a harmless, inactivated virus incapable of copying itself, but using very much the same strategy as Sarah talked about with how a virus itself works, because it is an inactivated virus. So our vaccine is a delivery mechanism. It delivers a little bit of coronavirus code to your cells, who then make the spike protein, which triggers your immune responses. And because we're delivering code, it's very easy to do that, as Sarah said, to re-engineer that to use it for a different pathogen. So to use it against rabies, or to use it against plague, or to use it against Nipah, or to use it against a new emerging pathogen. And it's relatively fast to do. And because the manufacturing step is going to be the same for all of those vaccines that we would make, because they're all inactivated or replication incompetent adenoviruses, you manufacture them in the same way, whether it's a vaccine against a new pathogen or against Nipah or MERS or rabies. The manufacturing process is the same. So that also saves us a lot of time. We don't have to figure out how to make it next time.
Now, you mentioned the mRNA there, and that is one of those things which I'm sure people who've ended up having kind of to-dos on social media may well have had that experience where people say, but that's not a real vaccine, so it doesn't count as a vaccine. So there seems to be this strange, I suppose it's that, is it because people still imagine, they look back and they imagine, well, there was cowpox and smallpox and that's how it's done. And that this is one of the first times that people have actually heard about the levels of technology that are being used in these new ways of combating viruses.
So there's lots. Another type of vaccine that we use is what's called a live attenuated vaccine. So vaccinia, the smallpox vaccine, is a live attenuated vaccine. It's very close relative of smallpox, but it doesn't give you the severe disease. But what that vaccinia vaccine is doing, that we don't need to use anymore because smallpox has been eradicated, that is a virus that's taking code into your cells to produce proteins that look very much like the proteins in the smallpox virus, so that your body makes an immune response against it. So this is nothing new. When Jenner started immunizing people with cowpox, which it's a long story, but we ended up using something called vaccinia, he's doing the same thing. He's giving people a viral vaccine which takes the genetic code inside their cells, and inside their cells you get the production of the proteins that you get the immune response against. mRNA vaccine just does it in a slightly different way. It's not a whole virus, it's just a piece of genetic information encoding one protein, the spike protein of the coronavirus in this case, and instead of being packaged in protein, it's packaged in a little bubble of lipid or fat, but it's essentially achieving the same thing. So going back to the very beginnings of vaccinology, the same thing was happening. I think people get a bit confused sometimes between vaccination and gene therapy, and say that whenever you're transferring in a gene, that's gene therapy. But these are genes that are only there for a very short amount of time. The messenger RNA doesn't hang around for more than a few days. The viral vaccines that we use don't hang around for very long either. There's no lasting impression. There's no, the genetic material doesn't stay in your body. So if you're trying to do gene therapy, transfer a gene into somebody to replace a defective gene that they have. That's a very different situation. There, you want the genetic material to stay long term, because it wouldn't be very effective gene therapy if it only lasted for 3 days. But vaccination isn't gene therapy, and the genetic material doesn't stay around for very long after the point of vaccination.
Right, and